liquid biopsy testing for mesothelioma holds promise as a less invasive alternative to tissue biopsies for detecting and monitoring this aggressive cancer. However, for patients to fully benefit from this technology, liquid biopsy tests for mesothelioma need to be standardized and regulated. Several key steps should be taken to help achieve this. Develop standardized collection and preparation methods. Currently, different laboratories use varied protocols for collecting, handling, storing, and preparing liquid biopsy samples like blood or urine for analysis. This introduces uncertainties and makes comparison of results difficult. Clinical studies are needed to establish reference standard operating procedures, SOPs, for collecting appropriate sample types, minimizing pre-analytical variables, and preparing samples consistently across testing sites. Adopting uniform collection and prep methods is crucial for obtaining reproducible and clinically meaningful liquid biopsy results. Validate analytical assay performance. Researchers are working to develop highly sensitive and specific assays capable of detecting the rare cancer-derived DNA shed into biofluids by mesothelioma tumors. Before liquid biopsy assays can be translated into clinical practice, their analytical validity must be established through rigorous validation studies. These studies should evaluate each assay's ability to consistently and accurately detect mutations known to drive mesothelioma, while minimizing false positive and negative rates. Performance criteria, such as sensitivity thresholds, specificity levels, and quality control measures should be set based on validation data. Conduct prospective clinical utility studies. In addition to analytical performance, the clinical usefulness of liquid biopsy assays needs demonstrated. Well-designed, prospective clinical utility studies directly comparing the results to tissue biopsy findings are needed. These should assess how well liquid biopsy detects mesothelioma, provides genotypic tumor profiling for personalized therapy selection, and monitors response or progression over time compared to current standard techniques. Data on how liquid biopsy may impact patient management decisions and outcomes would help validate its clinical utility. Establish standards for clinical validity. To issue recommendations on appropriate clinical use, regulatory agencies require evidence that a test provides clinically meaningful results. Clinical validity studies linking specific liquid biopsy assay results and corresponding mesothelioma case characteristics, for example histology, stage, prior treatments, must therefore be conducted. Statistical analyses should demonstrate the test's level of accuracy for intended clinical use case scenarios at defined performance thresholds. Standards delineating when positive or negative test results should guide clinical decision-making would promote standardized, high-quality care. Gain regulatory approval and oversight. Once sufficient data supports a liquid biopsy test's analytical and clinical validity for certain mesothelioma applications, the assay should seek regulatory review and approval. A pre-market approval process by the FDA would ensure assays meet specific standards for safety, efficacy, and manufacturing quality before being offered to patients. Ongoing performance monitoring after approval could help maintain high performance. Adopting a regulatory framework provides oversight that gives clinicians and patients confidence in liquid biopsy test results. Establish reimbursement pathways. Reimbursement policies that incentivize use of clinically useful liquid biopsy tests also help drive their adoption into routine care. Test developers will need to engage with payers to demonstrate a test's added value compared to existing modalities and cost-effectiveness. Studies quantifying potential savings from reduced invasive procedures or improved treatment responses could aid negotiations. Coverage policies that reimburse for liquid biopsy under defined guidelines when tissue biopsy may be precluded or impractical would promote standardized access for appropriate patients. Foster laboratory accreditation, ensuring consistent performance as liquid biopsy tests are performed across numerous clinical laboratories also requires oversight. Laboratory accreditation programs involving on-site evaluations, proficiency testing, and adherence to standardized procedures could help reduce inter-lab variability. Accreditation could be required to report results for clinical decision-making, providing an additional level of quality assurance. Continual performance monitoring may need to mandate improvements if necessary to maintain high standards. Promote clinical practice guidelines. Medical societies play an important role in guiding physicians on appropriate clinical applications of new tests. As the evidence base supporting liquid biopsy for mesothelioma matures, professional organizations should work to develop clear clinical practice guidelines. These guidelines would recommend scenarios where liquid biopsy has proven clinical utility based on established analytical and clinical validity data. Promoting standardized test ordering and result interpretation practices through guideline dissemination can optimize patient care. Overall, applying rigorous standards to establish the analytical soundness and clinical utility of liquid biopsy assays through well-designed investigations and demonstrating clinical validity will be key to support regulatory approval and implementation. Fostering accreditation, developing coverage policies, and disseminating practice guidelines can then promote consistent, high-quality use. Standardization and oversight through this comprehensive framework can help realize liquid biopsy's promise to transform mesothelioma management by providing a less invasive alternative or complement to tissue biopsy. Carefully conducted research and strong collaboration between stakeholders across regulatory, reimbursement, laboratory, and practitioner communities hold the greatest potential to standardize liquid biopsy testing for mesothelioma and translate this innovation into meaningful benefits for patients. Proper validation and oversight can maximize clinical validity and utility while giving physicians and patients confidence that test results guide care appropriately.